All right. Hello, everyone. My name is Michelle Ramsahoye. I'm a PhD student at University of Colorado Boulder, and today I'll be discussing the performance of graph neural network-based models for anti-CRISPR protein prediction. So my talk has three parts. I'll be talking about my motivation, which is can graph neural networks be used successfully for anti-CRISPR or ACR protein prediction. Then I'll talk about the process that I use to make that application. And then finally, what limitations exist for using GNNs for this purpose and how can I guide future research. So first, we have to introduce bacteriophages. Here I have Pseudomonas phage K8, and bacteriophages are virions that infect bacteria in order to replicate. So in this case, phage K8 will infect Pseudomonas aeruginosa. And we can think of these two as kind of being like on an eternal chessboard where they're constantly fighting against each other. As a defense mechanism, the bacteria has its CRISPR-Cas defense system, among other defense systems, where it uses it to recognize viral DNA and will eliminate it based on that recognition. And in response, phages sometimes have these things called anti-CRISPR proteins, which can then inactivate those CRISPR-Cas systems. So here I have a showcase of different structures of ACR proteins, and we can see there's 102 to date ACR proteins with 92 different subtypes. They are generally small, ranging about 50 to 150 amino acids. And they're diverse in terms of structure and mechanism. Here I have two ACR proteins, ACR-IF1 and ACR-IF2, and they both kind of function by binding to Cas, the Cas protein, to prevent DNA recognition. These proteins also tend to have low sequence similarity to other proteins. Additionally, ACR proteins have potential for multiple applications, ranging from reducing off-target activity of CRISPR, genome editing, detecting Cas complexes, or possibly being able to improve efficacy of phage therapy. So now that we have those three things in mind, they're small, they're novel, they have a lot of different structures, lacking sequence similarity to other proteins. I kind of thought so what tools and methods currently exist for ACR protein identification? And we can kind of divide them into both sequence-based and structure-based methods. So in 2013 is when the Bondi Denemy Lab first discovered the ACA or anti-CRISPR associated genes and their proteins. And then in 2021, literally I want to say May and then June, you had 3 labs come out with machine learning algorithms to classify or identify ACR proteins. And they were all mainly sequence-based machine learning methods. Then in 2022, 2023, we kind of see this shift of wanting to use more deep learning methods that are still relatively sequence-based. And just for clarification, when I say sequence-based, I'm really meaning they're using a sequence as an input. Generally, they're looking for some sort of homology or some guilt by association techniques, stuff like that. We also see in 2023, 2024 now a kind of shift into more maybe not sequence, but what if structure. So this 2024 paper I'm mentioning here is actually a wet lab paper showing, and there are a few others where the wet labs are actually more focused on looking at structure when it comes to finding or determining, characterizing these proteins, as opposed to using sequence. So somewhere in between all of my literature search, I came across a paper called Deep Fry. And this paper was a general protein function annotation and prediction model that used both graph convolutional network and then also combined it with an LLM to make predictions and apply gene ontology terms. And I thought this was interesting It was a way of taking both sequence and structural information and combining it together. And I figured maybe this could be a potential avenue for ACR proteins. So I had two hypotheses: that ACR proteins are not well predicted by larger all-encompassing sequence-based models. This makes sense because data is scarce. As I mentioned before, we only really have 122 characterized ones. And when you look at all the LLM-based models, they are being trained on the large, Interpro, Unipro, all of those huge protein databases, of which 122 is really small. And then likewise, by nature, these proteins have low sequence similarity anyway to other proteins. My second hypothesis was that GNNs, which can incorporate both types of information, can achieve higher prediction accuracies compared to models relying on sequence alone. And I say this mainly because graph data structures have been used previously for modeling proteins in different ways. There's the Amitai et al. Paper from 2004, where they used more network science-based techniques for predicting functional residues in proteins. Cannon et al. In 1999 used it for side chain clusters. And then likewise, I have this statement from a paper that talks about and says that evolutionary pressure tends to maintain structure more way after sequence deviates. So that brings me to this big thing that shows my process for using GNNs. And we can divide this into three parts: data curation, data transformation and labeling, and then the experiments themselves. So starting with data curation, I looked at a few databases commonly used for the anti-CRISPR models. This included anti-CRISPR DB. The Bondi Identity Lab has their own database that they use. And what we can see here in this tiny thing, this is based off the Gusau et al. Dataset, because I found it was most primarily used, is that when we want to use structural data, and by that I mean like X-ray crystallography, stuff that you'd find on Protein Data Bank, right? You don't have too much of it, right? You have mainly sequences with no known structure. No one's made the structure for it yet. So that means there's a gap if I want to use primarily structure. So I thought, okay, maybe we can use something like ESM-Fold2 or by Meta to make some predictions on these sequences to get a predicted structure. Likewise, as I mentioned prior, there are 92 different subtypes. And then when I looked at the benchmarking data set, which is again the Gusau et al. Data set, we can see that it's primarily type 1, type 2, and type 5, with others uncategorized. So when it comes to the other 89 types, then we can't really predict those. So next, I transformed both the experimental data that I had as well as the predicted structures using BioPython and Network X into what are called protein structure networks. And I followed mainly what DeepFry used within their own paper, which was they denoted the nodes as being represented as alpha carbons within each residue. And then an edge exists between two residues if the distance between them is within some threshold, right? The DeepFry paper used a threshold of 10 angstroms. I used 8 based on some more recent work. Next, I labeled attributes of interest to the graph. So I mainly focused on physiochemical attributes like dipole scale, volume. This is based off another model called AC Ranker that used these denotations. And then finally, I added an overall label to the graph itself, making this a graph classification task. Next came the experiments themselves. So I didn't really have a benchmark baseline model to work off of. So I varied some architecture, exploring different combinations of various hyperparameters like layers or number of neurons per layer. I did that for 2 types of models: the GCN or graph convolutional network, and then the GAT or the graph attention network. I then used k-fold cross-validation on the training set for each model, just to clarify each model architectures trained on the same data set or same training set before I tested them on the same test set. And finally, I have my kind of preliminary results. I have a training and validation loss curve here. The GAT, or the attention network, is in black, and then the convolutional network is in light blue. We can see that there's like a huge difference between the validation loss and training loss for the attention network. I'm thinking maybe this validation set is too small, maybe the model is too complex. But then when it comes to training and validation for the GCN, it looks something more akin to what we want to see. And then next, I have a few confusion matrices where I would denote the positive class as a protein being an anti-CRISPR protein or an ACR protein, and the negative class is a protein which is not an ACR protein. And we can see that between the two models, there's not really too much difference aside from the GAT having more of a tendency to have false positives, that is predicting something is an ACR when it is actually not. Next, we have the accuracy and F1 scores. Overall, the GCN has a slight advantage when it comes to accuracy and F1 score, just by a little bit. Accuracy of 89% and an F1 score of 91%. All that to say, there's a lot of limitations. Notably, we have a lack of experimental data, and I tried to kind of subvert that with some predicted structures, but also it's important to note that if you're using any sort of structure prediction to try to supplement your data set, any errors that come through that structure prediction can then also follow in any subsequent models that you use on it, right? So a good thing that I could follow up next with is maybe using AlphaFold just as a comparison, or also, I guess, comparing how well those models work. Then there's a lot of inconsistencies in the record keeping. Some of the data sets kind of vary between being an Excel file that just has a list of sequences that just say train and test, and then others have more explicit data telling me whether or not this has been experimentally validated. Is it just a putative ACR or something that we believe to be an ACR? Another thing to think about is the transparency of models within a biological context. Something that the Deep Fry paper did do was they included a part where they utilized their own kind of version of GradCam, which is a way of making generally a heat map on convolutional network images to find out what is the model focusing on, what's important. So there could be ways to kind of make the GNN models a little more explainable and map those results into more experimental data. And then finally, more wet lab verification. Out of all of the models, there was really only one, which was Deep ACR, that actually had an accompanying kind of wet lab procedure to validate the use of their model and how one would see it, or one would want a biologist or a biochemist to use their tool. And I think this is definitely an important part to consider when you're making machine learning models or any sort of tool. Is kind of how do you expect biologists or anyone to use them? That said, thank you for listening to my talk. I'd like to thank my advisor, Dr. Mirella Alistar, my prelims committee, and GLBIO for giving me a travel scholarship to come here present.
Thanks. That was super interesting. I have more of a biology question. Do you see these also in prophage inside the bacteria, or do you only see them in do they really get lost or purged when they're in the bacteria as opposed to just in the phage itself? And I was wondering also if there's any overlap between a while ago, like Amibot's lab had this thing about like all of these like small orfs in like bacterial genomes, and is there like any overlap between like these proteins and like those like unannotated small orfs?
Small.
Like small open reading frames, sorry.
Okay.
Yeah, like small, like, smaller proteins that are just like under 100 amino acids or so.
To answer your first question, yes, they do appear in prophages. Like I said, sometimes the data is kind of hard to get information out of. Sometimes they don't tell you what source those proteins may come from.
Yeah.
So it's really a matter of searching for that information. And then for your second question, you were asking if they...
Yeah, there's some kind of interest in like smaller like proteins that may have gone unannotated in microbial genomes just because like the typical tools tend to miss them, right?
Yeah.
And I was wondering if like any of those small proteins like overlap with the proteins in this set and like might have just been missed by like other annotation.
That is
methods.
A good question. Okay. I'm not sure.
Cool.
But thank you for bringing that up. I'll take note of that.
Cool.